Hello, and welcome to our final lecture in the Microbial Genetics lecture series, where we get to talk about mutations. And here we see some of the best known mutants in the world. X-Men. Even if you aren't a comic book fan, you probably know of Hugh Jackman as Wolverine and Patrick Stewart as Professor X. Might not know what they do, but you know that Wolverine has claws, and maybe Professor X is in a wheelchair. All right, but past that, they have these mutations that allow them to have special powers, right? And it's funny to think about that there's a single gene that gives them all these powers, a healing factor, and mental abilities, right? But it's also interesting to think about that it's kind of sort of real. Um, we don't get powers in the sense of like an awesome healing factor, but mutations do lead to uh, lizards and snakes being able to have some of their tails ripped off and then their tails growing back if they survived the encounter, right? Um, we grow back skin incredibly well. It does make scar tissue, so we don't grow back like hair follicles and stuff, but we do grow back some things and have something like a healing factor. And that came from mutations all the way back to the tree of life when we were um, related to a single-celled organism. And it kept mutating and mutating until we get to mammals, until we get to primates, and until we get to the divergence between the last common ancestor and humans. Okay, and so with that prelude in mind, we can actually see, okay, what are we actually talking about when we mention a mutation? So the most uh, complicated explanation is to just kind of look at this chart and make sure that you understand it, but let's go through it one by one, and that way we can kind of simplify it, okay? So before proceeding, mutations can be caused by either radiation, chemicals, UV light. Mutations happen in the normal DNA replication cycle. So anything can cause a mutation. Okay, most mutations, in the sense of in the DNA replication, will get proofread out and checked, and those will get fixed. But sometimes we are born with genetic mutations, right? And those genetic mutations are changes in a protein that we have. And these won't get fixed because each and every cell would have the same sequence. So they wouldn't get fixed because it's expected to be this sequence that we see. Okay, so remember we talked about genotype and phenotype, right? So this is the very heart of that. Okay, so we see our genotypes up here, and we see our phenotype here. Okay, all right. So genotypes here, phenotype here. Okay, so what the genetic code says, what we can see. Okay, what we see is that we can have silent mutations. This goes back to what we just did when we were testing ourselves with RNA translation. We looked that UAC and UAA were very, very close, and it was only a point mutation. But we saw that UAU and UAC were the same amino acid, and then we saw whole blocks coding for the same amino acid, all four codons coding for the same amino acid, just depending on which last codon we switch. Okay, and when that happens, that comes a point mutation, right? Because it happens at one point, right? That T, that one point, but it is silent. Okay, it's a silent mutation. Okay, it's a silent point mutation because in the normal type, the wild type we call it, it's a valine. Okay, And even though we changed this last amino acid, it is still valine. Okay, that is a silent mutation. Okay, because you don't see it in the phenotype. Okay, and we use the term silent even though we're talking about visuals. Just kind of bear with us, right? Okay. Then we can have a missense point mutation. Okay. What is a missense point mutation? It's kind of what we talked about also, except that when you have that UAC and it becomes UAG, it becomes a stop codon. We talked about that. And that is nonsense, okay, a premature stop codon. And we talked about that already. But the missense, I'm not overlooking, missense is the same thing, right? And instead of a stop codon, it's another amino acid, okay? So in one motion, we saw what nonsense was, and we saw what missense was, and we didn't even kind of put it together yet. Okay, so nonsense means that there's no sense involved in our amino acid. Okay, it prematurely stops. We're not going to have that protein most likely working, especially the sooner it is to the start. Okay, a missense will still make a protein. Okay, we'll still get a protein out of it. And as long as that amino acid doesn't throw it all out of whack, it doesn't like make a hydrophobic or a hydrophilic one instead, and it can still make the enzyme catalyst site, it might still work. Okay, it has a high chance of working, especially with just one point mutation. Okay, then you have insertions and deletions. When we talk about insertions and deletions, we are no longer talking about a point mutation. We are talking about insertions and deletions of masses of DNA. Okay, a prophage from a virus that integrates into the DNA would then essentially most likely cause an insertion where it sends all the codons out of frame and now makes a new protein. Okay, that's not unheard of. Okay, if that happens, it could kill the cell, obviously. So it's not likely to happen and still keep living, but it could still happen and does. Right? Insertions and deletions and point mutations. Okay, and I said frame shift. It's going to frame shift the whole codons out of whack. Okay, so these are the types of mutations you can have, and how we fix them is we usually have a DNA polymerase II come by if we make a point mutation in our DNA replication. But again, if we have an insertion or deletion, the DNA will just copy the other side, thinking that we forgot to do the other side in DNA replication. Okay, so we can fix some mutations. We can fix point mutations during proofreading steps, but we cannot really fix genetic mutations just yet. There are some things that we'll talk about in the DNA Tech chapters that do this, but they're not available just yet.